Thank you very much. Well, ladies and gentlemen, certainly we are expecting to join more interesting discussion with all speakers today. And now, we will start the second day of GAMA-ICTM 2022 with the plenary session 2 under the topic of One Health. And this session will be led by our moderator, Professor Wibawa from Universitas Gadjah Mada. Here it is the profile of Professor Wibawa. Professor Wibawa is a professor at Department of Microbiology, Faculty of Medicine, Public Health and Nursing, Universitas Gadjah Mada. He graduated as medical doctor from Universitas Gadjah Mada, and his doctoral degree was earned at Kobe University Graduate School of Medicine, Japan, in the major of molecular medicine. Then Professor Tri continued the clinical microbiology specialist in Universitas Gadjah Mada. His research interests are antimicrobial resistance, genetic susceptibility of infectious disease, drug development for infectious diseases, and vaccine development. Well, ladies and gentlemen, please welcome Professor Tri Wibawa to lead the session.
Mbak Dr. Eka, Assalamualaikum warahmatullahi wabarakatuh. Good morning, all participants. It is a great honor to welcome you back to the second day of the Gadjah Mada International Conference on Tropical Medicine 2022, which will be held November 15 until 17 virtually. My name is Tri Wibawa. I'm your moderator for this morning plenary session. As we know, One Health is an approach that recognizes that the health of people is closely connected to the health of animals and our shared environment. One Health is not a new concept, but it has become more important in these recent years. This is because many factors have changed interaction between people, animal, plants, and our environment. One Health approach has been formulated in many actions plans as the best approach to tackle many problems in many issues. In relation with the importance of One Health approach in this tropical medicine area, we bring you the plenary session for second day with One Health session. Within this session, we will learn from three prominent scientists that has been working in One Health for many years. And we are welcoming three speakers. The first speaker is Professor Wiku Adisasmito from University of Indonesia. His talk will be about health security related to One Health concept. The second speaker, Professor Roger Frutos from CIRAD. He will talk about the Zoonoses and One Health concept. And the third speaker will be Dr. Matthew Grigg from the Menzies School of Health Research. He will talk about the One Health approach for zoonotic malaria. And for your information, we are not going to provide you the question and answer session for this plenary session. And ladies and gentlemen, let us open our plenary session. Please, Prof. Wiku Adisasmito, you may deliver your talk during this coming 20 minutes. And before I will introduce Professor Wiku Adisasmito. Professor Wiku is a spokesperson for the Indonesian National Task Force for Food and Mouth Disease, or FMD. He's a commissioner for Kimia Pharma, and then he was awarded as Pahlawan Inspiratif 2021 by the Radio Republik Indonesia, or we can say Inspiratif Hero Award in 2021. And then let us welcome Professor Wiku. Please, Professor Wiku, 20-minute time is yours. Thank you.
Oke, jadi mungkin saya mulai saja langsung ya. Saya harus menyebutkan siapa saja ininya yang ada di situ atau enggak? Langsung isinya atau gimana? Oke, ya sudah. Ya udah, saya langsung saja ya. Oke. Good morning. Thank you very much for inviting me to the Gadjah Mada International Conference of Tropical Medicine, and with the title Topics of Health Security Related to One Health Concept. And as a government spokesperson of COVID-19 Task Force and also as a professor at the Faculty of Public Health, University of Indonesia, I would like to share some of the knowledge and experiences about the health security related to One Health concepts. First, next slide. I would like to show you that there are challenges that we face globally and also nationally that because of the several disruptions of the land use change, global mobility, man-made disaster, climate change, natural disaster, and also infectious disease outbreak like COVID-19, we need to, as a world, we need to make sure that we can respond and learn from what we are facing lately with the pandemic of COVID-19. And of course, this will be beneficial for all other disruptions causing by the emerging infectious diseases. Next slide. And if we look at the national experience in Indonesia, there were several incidences in Indonesia that potential misuse of biological agents. First is in 2012, where the French embassy received a shipment suspected of containing anthrax powder. And also that in 2015, there's also with the anthrax, the other embassy, which is the Australian embassy, is also disrupted with this issue. So there are other issues that we are facing with the emerging infectious disease or biological agents that related, which cause disruptions to the human activities. Next slide. And I would like to share with you all about the disease threats that we are facing, which I call it disease of tomorrow. Imagine this happened in November 2019, which we don't know whether that's going to be SARS-CoV-2 outbreaks. So that's, imagine that's the time horizon, that at that time we don't know what's going to happen to tomorrow. It's the same thing when we are sitting now in November 2022. We need to make sure that we should know that what would be the disease of tomorrow. Tomorrow meaning probably early next year, or hopefully there will not be happening of something like a threat like this in the years ahead. But because of the imbalance of nature, because of the interactions of human, animal, environment, and also food, we want to make sure that we know what's going to happen. Look at this threat that after the first publications in 2004, in the last 18 years, there are 5 infectious diseases importance that becoming global threats and realizations into problems to all of us. So we need to make sure that we are able to detect and respond if there is something happen in the future. Next slide. The world basically had understood the situation. In 2014, several countries launched the Global Health Security Agenda. And if you look back in 2014, we already identify several key issues related to the prevent, detect, and response. And we developed all together with the members of the countries in the world, 11 global health security action packages, including antimicrobial resistance, and other topics where it's important. And some of those, including number 5, national laboratory systems, real-time surveillance, number 6, reporting. Those are the issues that we were facing at the beginning of the COVID-19 outbreak in Indonesia and also globally. We face the problems where the laboratories are not connected, but finally we can connect those of the laboratories to improve the ability to detect where the virus is circulating. So this is one of the issues that we learned, that although we understand, we know that it's global health insecurity in the future, but we could not predict at that time. And then we responded relatively late to the COVID-19 all over the world, not only in Indonesia, but all over the world. So this is a lesson learned, and we should not repeat the same mistakes in the future. Next slide. Therefore, I would like to share with you the One Health approach to combat the disease of tomorrow. Since the disease of tomorrow relatively related to emerging infectious diseases, which originated either it's from human to animal or vice versa from animal to human. So we need to make sure that the national health system or national human health systems are also interconnected with the national animal health systems. Therefore, if it's interconnected and we are structurally established in all countries, including Indonesia, I'm talking not only in Indonesia, but it's all over the world, should have these systems established from local, which is at the, in Indonesia, we can say it at the level of the city and district level, up to the national level, regional level, and global level, that we work together. So the system itself, health system itself has to be interconnected and work across and with the principle of the One Health, which is connecting human, animal, and environmental issues, where, of course, in this issue, we want to make sure that the thinking, it should be a system thinking process, and leadership, not sectoral leaderships, but multi-sectoral leaderships, and also communications and informatics, which is sharing the data, etc., where we are sitting on different values and ethics of the sectors, and that's the challenge, so that we need to be integrated and systematic approach to make sure that we have a resilient to the disease of tomorrow. Next slide. And for this issue, we want to make sure that One Health collaboration should happen, as I mentioned, at the global level, regional level, national level, and local level. And we know at this moment, the quadripartite, WHO, FAO, UNEP, and also WOA, which is World Organization for Animal Health, are working together, and they have appointed 26 experts across the world to give advice to this quadripartite to make sure that the basis of the scientific evidences, which should drive the advice to those quadripartite in responding or preparing or preventing the emerging infectious disease, which may lead to the pandemic in the future. So that's therefore this is an important issue for us to consider, that the One Health approach has been considered at the global level in the UN bodies, and still the challenges in the future is to make sure this is also organized in collaborative manner at the regional level as well as at the national and local level. Next slide. And the One Health high-level expert panels, which advise the quadripartite, have developed the One Health theory of change, meaning that the way we manage the world, which need to collaborate across sectors, human, animal, environment, and also food, and as an integrated efforts, need to make sure that we can make the change so that the world managed better at the global level, regional level, national level, and local level. We cannot only talk at the global level without considerations in the implementation level, which is at the local level. So the governance, the global governance is so important, and the pathway of change has been identified and verified with the quadripartite. And of course, this is a platform of collaborations where we, the sectors and the organizations at the global level discuss together to make sure that we can achieve the outcome better. Okay, next slide. Learning from the experience in Indonesia, next slide. We strengthen the health security using the 5S1T approach. I'm sorry, please, next slide. Okay, thank you. So there are 5S1T, which is basically, this is the approach that we developed in Indonesia in responding to COVID-19. And I think the 5S1T is also relevant to other emerging infectious diseases. So first is to make sure that we have the strategy. Prevention is much more important than curing. So we need to have the structure from central to local with the community-based and pentahelix approach. We need to mobilize all of the resources and stakeholders. We work in the systems, make sure that the systems, the organization systems work well. And then if we are struck with the something new like COVID-19 at the beginning, we don't know about what is this, we need to have the skill. We need to have the expertise, we need to train, and we need to quickly improve the skills to make sure that when we react, when we work together in a high speed, because we need to protect the human beings, the animal, the also the plants and others, because that's an ecosystem that need to be balanced. Next slide. Back to the COVID-19, that lessons learned in Indonesia, that the cross-sectoral coordination is so important, where we connect the central government with the local governments. Everybody understand everywhere in the world the decentralizations, because part of the democratization, each area or regions has its own authority, although they are in one country. Sectoral variations in provinces and district level, causing obstacles when we are facing with the pandemic like COVID-19. So therefore, Indonesia is making a task force where the task force itself incorporate the civil servants, civil organizations from Ministry of Internal Affairs from the central level up to the local level at the neighborhood level. And then at the same time, we mobilize the army and also police who already have the command systems from central to local. By connecting those infrastructures, therefore, the actions from central governments, knowing that there are some decentralizations and local authorities, this can bypass the system better and move quickly, as I mentioned before. And then at the local level, we mobilize the stakeholders. So this is not a concept, what I mentioned to you in the slides. This is a reality. This is what we are doing for 2.5, almost 3 years right now. And then we have 4P, which is prevention, management, development, and supporting functions at the local level. And it's monitored centrally, real time. We receive the reports of people activity in terms of health protocols, in terms of vaccinations, real time and interoperable. So the data can be seen by all levels. Next slide. So if we have this type of approach by pentahelix collaborations in policy, regulations, I'm involved in developing the circular of the task force, where we regulate the people mobility at the domestic level as well as international level, where people mobility in and out the country and inside the country. I really consider all of the evidences, the dynamics of the COVID-19 in other countries as well as in Indonesia, from central to really district level. So we have all of the data, and we mobilize the government, the media, the private and academicians all together, because we have to combat this disease or this problem together. And I think this is a good investment for a country like Indonesia, and of course to other countries, to make sure that we can survive and we can win with this pandemic or emerging infectious disease. Therefore, health security can be improved if one country implement like this. So all countries implement like this in concerted effort, and it's organized globally with a quadripartite at least. Then the world, I think, is more secure and more transparent, because the threat can be identified and can be mitigated before it causing a lot of problems. Next slide. And for that, we need to prepare the response to public health emergency. This is like if something happened. So that's why we need to prepare first, and we have the early warning, because we share the data, and this is a big challenge in the world, that we need to share the data not only from the human health side, but also from the animal health side, from the environmental side, as well as and the food system sides. And it's also 4 of those are interconnected, work together. It's a big challenge. It's easy to explain, but it's hard to be implemented, because naturally we are grown in a system where it's very sectoral. But now we are challenged to work together. So that's why One Health is an important approach to make sure that the global health security can be maintained. Next slide. And I would like to mention about the capacity to detect, which is based on the laboratories. At the beginning of the pandemic, we rely on the laboratories from the Ministry of Health. And then finally, we realized that we cannot have a high speed of identifications, detections, if we only rely on several laboratories only from the Ministry of Health. So then we make sure that we mobilize all of the laboratories of life sciences that is important for detections of COVID-19. And I think these laboratories can be used for other purposes in the future if we are challenged with these emerging infectious diseases. Of course, the practices in biosafety, biosecurity need to be improved of all of the laboratories and interconnected. So the human resources interconnected, the facilities, etc., is also connected, and the information and data. Next slide. But the challenge still remain there. You know, I show you here that there are almost 20,000 life science laboratories in Indonesia, which is under the different authorities, whether it's under the military, under the Ministry of Education and Culture, under the agriculture, etc., and including the health. So how can these laboratories are interconnected in one concerted effort, where we know the human resources working there in a different sectors, the facilities are planned differently. So therefore, this is a big challenge for us. But once we can interconnect them, at least for COVID-19, the efficiency in detections improve quite a lot. And I think for the future, even with the foot and mouth disease right now, which is only the problem of the animal health, but we try to use laboratories that exist for the COVID-19. And still there are some problems involving that, because the people in the laboratories also, or the sectors, still think that that's an animal health issues. It's not human health. If for human health, then the animal health and other laboratories need to support. I think that's unfair, looking at the facility from the perspective of the human health. One Health approach is comprehensive, and it should be at least from the 4 sectors: human, animal, environment, and also food system, which interconnect all of those. Next slide. And for that, we need to make sure that we improve the biosafety and biodefense, because the risk of the biorisk, it can be originated with at the natural level, accidental, and sometimes intentional. And we need to avoid to happen on the right side, so intentional. So therefore, we need to have the biosecurity systems interconnected. Indonesia has been investing in that rather slow, but it's better compared to other countries. And we need to improve that in the future to make sure that we are ready to respond and to prevent something happen in the size of COVID-19 pandemic. Next slide. Indonesia is trying to regulate the select agents. So we need to know what are the select agents, which is dangerous for the environment, for the human beings, and also animal. And then we need to know where those collections are stored and managed by whom. And we want to make sure that the use of those agents are for the positive uses. And we need to make sure that we regulate that and monitor that. Next slide. To incorporate all of those, since the start in 2018, the Indonesia One Health University networks and working with other stakeholders try to sensitize the government of Indonesia. Again, like the Global Health Security Agenda, as I mentioned earlier in 2014. And then also this 4 years after that, Indonesia is through the university network initiated the effort, but with the commander of the armed forces, we want to make sure that we need to have the biosafety, biosecurity, or biodefense, biosecurity and biodefense bodies, which incorporate all of those laboratories and facilities when in the era like COVID-19, we improve the capacity to detect, we improve the capacity to develop vaccines, we improve capacity in responding to the emerging infectious disease like what I mentioned. So One Health approach, one of the approach is multi-collaborations across institutions, across sectors, and from central to local, if we are talking about national, from global to local. Again, it's a task for all of us, including academicians, to connect the global level, the regional level, the national level, and the local level. So next slide. So I would like to say thank you for your attention, and I hope this inspire all of you that we need to improve the global health security by improving the governance from central, from global to local, using the One Health approach. So thank you very much for your attention, and I hope this is important for all of us. Thank you. Selamat pagi.
Yep, thank you, Prof. Wiku, for the presentation. I'd like to highlight several points that has been mentioned by Professor Wiku. There are several diseases that should be recognized as potential threat for the future. And then One Health approach has been proven to be a potential tools to combat with this disease of the tomorrow. And it was one of the lessons learned that is already learned from the COVID-19 pandemic, especially in Indonesia. Thank you very much. And then we going to welcome to the second speaker, Professor Roger Frutos from the CIRAD. And I'd like to introduce the second speaker, Professor Roger Frutos is the director of the CIRAD, where he specializes in infectious disease, dynamic, and infectiology. He is also the Association of Southeast Asia Institution of Higher Learning's Director of Project and Partnership. And he has around more than 130 articles published in international journal, and it is related with a One Health approach and with it's index 38. And then please, Roger Frutos, you may deliver your speech for the next 20 minutes. Time is yours, Prof. Frutos.
Ladies and gentlemen, good morning. It is my pleasure to be with you today. And first of all, I would like to forward my gratitude to Universitas Gadjah Mada and to the organizing committee of the GAMA-ICTM 2022 conference for inviting me and for giving me the opportunity to give you this presentation. My talk today will be on zoonoses and One Health. We all know today that zoonoses are an important issue, especially after basically 3 years of COVID-19. But what are we talking about exactly when discussing about zoonoses and One Health? When we mention the word zoonosis, we immediately think about diseases present in the wildlife, which are moving to the human population. So therefore, when talking about zoonosis, we are automatically talking about emerging infectious diseases emerging in the human population. But what is it exactly? We need to go into some exact and accurate definition to understand what we are talking about. So what is a disease? First point, first question. A disease is a pathologic, physiologically abnormal condition. It's a deviation from a normal, or in other words, healthy condition. Yes, but what does it mean? And we still have the same question. What is a disease? How do we define a disease? A disease is defined and identified by a pattern of descriptors, a pattern of symptoms, in other words, a syndrome. With no clear pattern of symptoms, a disease cannot be diagnosed, cannot be recognized, and the physician can do nothing. This is the very nature of medicine. Medicine is syndromic by essence. If there is no recognized symptoms, there is nothing that can be done. And that is very important in the situation we are talking about. Because what is an infectious disease? An infectious disease has an ecological dimension. Why? Because it is caused by another living organism. And each living organism in this world, on this planet, is impacting its environment. Each living organism is damaging its environment during its life because of its life cycle. The problem is that the environment of a parasite or a pathogen is another living organism. In the case of medicine, it's very simple. It is us, humans. And the damage caused by the life cycle of these living organisms are what we call the symptoms. And the effect of that is what we call a disease. So it's really important to consider what is the real dimension of a disease. And we have to consider such that zoonosis does not exist. It's an intellectual construction. There is no transfer of diseases from animals to humans. They are instead multi-host pathogens that can therefore move from one host to another, They are capable of infecting different species, and in each species, they will trigger a different disease, because in each disease, you're going to have different set of symptoms, different patterns of symptoms, different syndromes, therefore different diseases. It is not the disease that is moving, it's the pathogen. So the dynamic we have to address is that of the pathogen. But before going to that, we need to consider a very important issue, which is the issue of the anthropocentrism or homocentrism. This is a very essential issue in this domain. We have in the past something that has been called the Copernican revolution, and we can refer to that. That was in the end of the 15th, middle 16th century, when Nicolaus Copernicus developed a new model different from the Aristotle's universe, or also called the Ptolemaic universe, which was geocentered. What it means is that the Earth was considered the center of the universe, and everything, all planets, all stars, and the sun were moving around, revolving around the Earth. And this is simply a consequence of the observer bias. Why do we have this bias? Simply because every morning we see the sun rising in the east, going up at noontime in the sky, and going down and vanishing in the west at sunset. So the obvious conclusion from that is simply that's because of the sun is revolving around me. But it's an observer bias, which is what Nicolaus Copernicus showed and said, no, it's because of the relative movement of the different planets and the Earth and the Moon and the Sun. We have a different movement, and it is actually heliocentered. The sun is in the middle, not the Earth. The Earth is revolving around the sun. Simply because of this observer bias, we have the impression, we have the feeling that things are revolving around us. But it is only a bias. It is not the actual physical situation. We have exactly the same problem when it comes to emerging diseases. We consider the human species as the utmost element of the evolution. We are really the final stage, and everything is revolving around us. And when it comes to a disease, it is necessarily a disease that comes from animals and will go to humans, either directly or either through an identified species or putatively identified species. I'm taking specifically and on purpose the example of COVID-19 because everybody knows it, or a putative, not yet identified intermediate species. But there must be a movement of the disease from the animals to the human. Why? Again, because of this observer bias, because we are not affected by the disease. All of a sudden, we are affected by the disease, so it must come from outside, and it came to us. And there is a bias in that, because what infectiology has to do is to make its Copernican revolution. In other words, we have to move away from the anthropocentric vision. We have to do the same as Copernicus and consider that what is in the center is not the human population, is not the human. What is in center is the pathogen, whether it is a virus, a bacterium, a parasite, is the pathogen, which has the central role in this system. And what we are experiencing is just the dynamic of the pathogens we can, in fact, infect different species and move from one species to another in different reactions. Human can be infected by pathogens coming from other species, but in turn, other species can be infected by pathogens present in humans. So we have to move away from this homocentric or anthropocentric vision to really understand what infectious disease and emerging infectious disease is. And we have to really identify who is the real master. The real boss is the pathogen. Is the pathogen which has the active role. Well, I'm taking on purpose again the example of COVID-19 because it's fresh in the memory of everybody, and it is easy to understand, therefore. So I'm going to talk about the virus model, but you can translate it in other kind of pathogens. So we often see the virus as passive. Often it is referred to as a propagule, therefore some passive element which is transmitted from one person to another, one host to another, just like a coin in the pocket that moves from one pocket to another. But it's not true. It's completely wrong view of the system. It is not passive at all. It is actually the active one, because these viruses, like SARS-CoV-2, are host switching machines. They are multi-host by nature. They are evolved for that. They have adapted for that. And these viruses, like all viruses actually, are highly evolved. The passive form actually is the host, is us and other animals, because the viruses are using the host for both replication and propagation. So what do they need for that? Oh, it's very simple. They don't need much. And we need also to clear some misconception about that. Viruses recognize receptors or docking sites, more precisely, because the receptor is something different. They are recognizing docking sites prior to entering the cell, okay? Multi-host viruses, like SARS-CoV-2, recognize receptors which are present in many species. It's the same receptor shared by many species. But what are those receptors? They are simply proteins which are essential for the physiology of the host. The host needs it for its survival, okay? And it's an evolutionary trait from the virus. They have evolved to be able to recognize these receptors present in different species. These receptors are generic receptors, and in other words, they are receptors which are core proteins encoded by core genes, and they are deep core protein, in other words, present in many, many species. In the case of ACE2, which is the receptor recognized by SARS-CoV-2, it's present in all vertebrates. So the virus can recognize potentially the receptor in any species, okay? What they need is simply contact and a minimal affinity for the receptor, which means that even though it is present in all species, the virus cannot recognize all the receptors because there are some variation depending on the species. And sometimes the virus will be able to recognize it and then to bind to it. And other cases, the virus will not be able to recognize it and will not be able to bind to this receptor. And I mentioned that there's a second point to consider to understand really the process is that viruses evolve, and they evolve very fast. And that's another key issue. And they evolve, if we refer again to the pandemic virus, to the emerging viruses, and to the coronaviruses, for instance, they evolve through a very specific model of evolution, which is called the quasi-species model, and which is specific to RNA viruses. Why? Because in RNA viruses, the replication is made by a protein, which is called the RDRP, or RNA-dependent RNA polymerase, which is a very unreliable polymerase. It makes a lot of mistakes, 2 mistakes to be viable. So there is a big problem because RNA viruses do exist. So how come they do exist with just a very bad replication system? The reason is that they have a very specific model of evolution and adaptation, the quasi-species. Basically, it means that the population of viruses display all possibilities of mutation, not in a single variant, but as a whole population. Each variant carries only up to 3 mutations. That's it. But most of them, all of them together, they explore the whole possibilities of mutations. And most of them are zombies. They are not variable. They will be created, they will be made, and then they will disappear immediately. However, here and then, some variants will carry mutations by accident, which allow them to overcome the host defense and therefore to replicate. And those will be selected and will make the basis for a new generation. And that's the way these viruses can move from one host to another and can adapt also to the evolution of the defense system of the host. And to say it clearly from now on, that's how also they adapt to vaccines. So it's very important to consider that. So basically, and that with one exception, it's not very clear, all human emerging viral diseases are due to RNA viruses. It is not a coincidence. It is not a mistake. It's because of these specific traits of RNA viruses to evolve through a quasi-species system and adapt, and to recognize generic receptors. If we go very briefly on the quasi-species of evolution, its system, it's very important to spend maybe a minute on that, because it will help us a lot to understand what we are talking about after that. If we start from one single variant, okay, one particle, which means one virus, one genome. This virus will infect a cell, recognize the receptor, be able to replicate, and will generate a very large population of virus. So one virus infecting a cell can generate 10 to 100 viruses per infected cells, with 2 to 3 mutations per genome in the average. But this means also 1 to 100 billion viral particles in a single human being, okay? And this virus will, as we mentioned, adapt to the host defense, because they will be able to overcome it. It's a host-driven selection. And this has a very important consequence, because if you change the species, in species A, you will have a given pressure of selection, because the immune system is specific to that species, and the virus will evolve in a given way. So it will give virus A specific to species A. In the species B, the conditions will be different, and it's a different set of mutations will be selected, and you're going to have a virus B. In species C, again, the environment will be different, and other mutations will be selected, and you will have a virus C. So in each species, the evolution of the virus will be different. So we have several major conclusions to understand from that, which will be very useful to understand the rest of the presentation. First thing we have to understand clearly now is that this specific mutation, in other words, in our case, the mutation specific to human, the human signature, are occurring post-infection, not pre-infection. So it is useless to look for these mutations in the wild. They don't exist. They exist only after having infected humans. And what is One Health? One Health is exactly that. It is to consider the true position of humans, not to rely on narratives or imagination. We need to consider the true dimension of the pathogens. They are the active component. The true understanding of their dynamic, how they evolve, and how they can move from one host to another and adapt. And the key role of society and human pressure in the emergence of propagation of infectious diseases. That is really One Health. Understanding our real position and act accordingly and move away from the bias of the observer. And with this, I will end this presentation and thank you for your attention.
Yep, thank you very much, Professor Frutos, for your lecture. And then I'd like to give several highlights from the Professor Frutos' talk. He reminds us that we need to consider again about the zoonotic concept, because before we are using the anthropocentrism, and then he offering us to reconsider to make a revolution to become a pathogen concentrism. So as a model of this pathogen concentrism is that Professor Frutos has been highlighted about the pathogen model of evolution. And then we know that actually after several times learning from the pandemic situation, we know that almost all emerging viral disease that is become pandemic is actually the RNA virus that is can be understanding by the pathogen concentration. Thank you very much, Prof. Frutos, and I will go to the third speakers. I will invite Dr. Matthew Grigg, and that is going to talk about One Health approach for the zoonotic malaria. Let me introduce the third speaker, Dr. Matthew Grigg. Dr. Matthew Grigg are Principal Research Fellow, Associate Professor at the Menzies School of Health Research in Australia. And he is now remote medical practitioner at Miwat Aboriginal Health Corporation in Australia, and he has been awarded as a Allen Plant Memorial Prize in Infectious Disease Epidemiology from the UNSW and Department of Health Australia government. Dr. Matthew Grigg will deliver his talk for 20 minutes. Please, Dr. Matthew Grigg, time is yours.
Hello, everyone, and thanks for the opportunity to speak today at the Gadjah Mada International Conference for Tropical Medicine. My name's Matthew Grigg. I'm a Principal Research Fellow and also a medical doctor based at Menzies School of Health Research in Australia. It's my pleasure to talk about one of my favourite topics, which is zoonotic malaria, particularly with a focus on monkey Plasmodium species originating from Southeast Asia. So just a quick overview of the talk. I'll give some context for monkey malaria in general. Particularly the focus of this talk will be on the zoonotic parasite Plasmodium knowlesi, and I'll discuss some of the history and origin around this species. I'll also discuss the geographic distribution of Plasmodium knowlesi across Southeast Asia, some of the diagnostic challenges which really underpin our lack of understanding about the transmission extent and disease morbidity. Importantly, some of the environmental drivers of why we might be seeing an increase in cases, and some of the integrated multidisciplinary ways we could approach this complicated transmission cycle. So a quick context and overview on monkey malaria. Non-human primates are known to have over 30 Plasmodium species, which can infect a variety of monkey and apes. In Southeast Asia, the main focus is on the 5 major species which infect the long-tailed and pig-tailed macaques, being Plasmodium knowlesi, P. cynomolgi, P. inui, P. fieldii, and P. coatnei. In South America, however, there's some other interesting species, P. simium, which is closely related to vivax, and P. brasilianum, which is closely related to P. malariae. However, across the globe, most species of Plasmodium are host restricted, and this sort of defines often their geographical range. Plasmodium knowlesi is unique in terms of it is known to cross multiple host families, both across monkey species and also obviously humans, and experimentally even a much wider range of monkey hosts than what happens naturally. So a quick overview on the evolutionary history of Plasmodium knowlesi. So it's been around for a long time in Southeast Asia, more than 100,000 years ago. There was a large population expansion of P. knowlesi at the same time as there was a concurrent increase in the human population. There was divergent subpopulations which spread from mainland Southeast Asia to the island of Borneo about 13,000 years ago. However, it wasn't until really the early 30s that the most detailed morphological and descriptions of Plasmodium knowlesi were made by Knowles and Dasgupta in India based on a blood smear from a long-tailed macaque imported from Singapore. And that was the time when P. knowlesi was officially named. It was quickly adopted as treatment for tertiary neurosyphilis at the time due to its fast 24-hour cycle, which the fever effectively treated the syphilitic infection in the brain. However, it wasn't until the 1960s that the first naturally occurring infection was reported in humans. This was present in a US Army surveyor in Peninsular Malaysia, who on return to the US had his infection confirmed through passage through monkeys. This sort of really triggered a huge amount of interest in how common zoonotic malaria may be across Southeast Asia. A couple of large surveys at the time really didn't show too much, and I think that was predominantly because they were constrained by the limitations with the detection tools at that time, which was actually just inoculating monkeys to see whether they developed fulminant infections. So soon after that as well, it was experimentally proven that transmission from infected mosquitoes could transfer P. knowlesi from human to human and also from human back to monkey. However, it wasn't until 2004 that Professor Bal Singh, based in Kapit in Sarawak in eastern Malaysia, described using PCR confirmation a large focus of naturally occurring P. knowlesi infections in humans. And there's obviously been a huge amount of interest and resurgence in studies focusing on not just P. knowlesi but zoonotic malaria in general since that time. Broadly speaking, the distribution of Plasmodium knowlesi reflects the geographical range of both its primary Macaca fascicularis and Macaca nemostrina monkey hosts, which overlaps with the Anopheles lucus virus group of mosquitoes. And now pretty much all countries throughout Southeast Asia where these the monkey hosts and mosquitoes exist have now had PCR confirmed cases of human malaria due to P. knowlesi infection. A key reason for the lack of understanding about P. knowlesi transmission prior to 2004 was that current conventional point-of-care diagnostic tools for malaria, such as microscopy in particular, weren't able to detect P. knowlesi. So the morphology of the parasite is similar, almost basically identical to P. malariae, and the early ring stages are also identical to P. falciparum. So on routine microscopy, most studies have shown that they're commonly misidentified. Particularly misdiagnosis with P. malariae can have important clinical implications, both to delayed appropriate treatment and recognition of the severity of disease has led to case fatalities in Malaysia in the early part of our understanding about what was happening there. Other common diagnostic tools, such as rapid diagnostic tests, which are based on parasite lactate dehydrogenase detection, have proved insufficiently sensitive in the earlier iterations of these tests to detect P. nolzi. None have been specifically designed to detect P. nolzi, and we rely on cross-reactivity with, for example, P. vivax, PLDH based tests. However, on more recent rounds, we have seen an improvement in the sensitivity, which now for parasite counts above 200 actually exceeds 95% for the best performing RDTs. But in reality, we really need highly sensitive and accurate molecular tools such as PCR to definitively confirm P. nolzi infections. So why is P. nolzi actually a public health threat? Well, the story of Malaysia, which has been the most studied area for P. nolzi to date, really highlights that as other cases of P. falciparum and P. vivax have come down in response to traditional control efforts, the increase in cases due to P. nolzi has risen. It's particularly highlighted, for example, in Sabah in East Malaysia, where the number of cases exceeds 2,000, all PCR confirmed to date, and we have seen quite distinct heterogeneity in the areas of higher transmission, for example, along the Crocker Range. Importantly, it's not just an increase in cases, but the fact that P. nolzi is not a benign parasite. Severe disease occurs in about 6 to 9% of patients, and case fatalities occur about 2.5 out of every 1,000 cases in Malaysia. Importantly, current malaria prevention activities may not be as effective given the monkey reservoir. Importantly, the recent WHO Malaria Policy Advisory Group has advised that in areas where hundreds or thousands of P. nolzi cases are present, certification of malaria elimination should be postponed. So just to give a bit of a broader overview on the P. nolzi biology and the life cycle, similar to other Plasmodium species, initially after being bitten by an infected mosquito, the sporozoites go first to the liver. There's no dormant hypnozoite liver stage in P. nolzi, so no need for radical cure with primaquine. The blood stage asexual cycle is fast, as I mentioned, 24 hours. It takes about 8 to 12 days before people become symptomatic and have a detectable amount of parasites circulating in the peripheral blood. Similar to P. vivax, P. nolzi uses the Duffy antigen to invade predominantly young red blood cells. However, for cases where we're seeing much higher parasitemias, there are alternative invasion pathways such as binding of normocyte binding proteins in older red blood cells also. Gametocytes are present in the majority of infections, which has implications for the possibility for onwards transmission from human infections. And it's well established, obviously, that monkeys can transmit to other monkeys and also to humans, but one of the major questions continually has been whether human-to-human transmission is occurring. As I stated before, it has been proven experimentally in the lab. From an epidemiological point of view, we have seen peridomestic transmission with some family clusters, including with women and young children, suggesting that it's a possibility. However, a lot of the ways that we've tried to understand this to date haven't shown any strong evidence that this is occurring. For example, analysis of genetic sequences from P. nolzi parasites derived from human and separately from monkey infections haven't shown any completely separate haplotypes, indicating that there hasn't been a divergence in terms of separate infections between the two hosts. Mathematical modelling as well suggests that human-to-human transmission scenarios where that could happen is very, very low probability. We've also found no evidence of drug resistance, which we would expect to see, for example, with sustained human-human transmission with treated infections. Importantly, one of the interesting features that may be indicating an increased rise in cases, for example, in Malaysia, is the the presence of cross-protective immunity to P. vivax. This has been shown nicely in a couple of studies now where certain P. vivax antibodies will protect against P. nolzi invasion in red blood cells. In areas where P. vivax transmission is being eliminated or close to elimination, that waning cross-protective immunity may mean that people are at higher risk of developing symptomatic P. nolzi infections. So what about in Indonesia? Importantly, based on risk mapping incorporating land use change, the presence of macaques and the relevant mosquito vectors, highlight there are a number of areas that where there's at-risk populations. We've now seen PCR reported cases of P. nolzi in Aceh, in Sumatera Utara, and also across Kalimantan now as well. What about other zoonotic species? So importantly, P. cynomolgi, which also is present in often in co-infections in the long-tailed and pigtailed macaques. It does have a dormant liver stage similar to P. vivax, and we have seen PCR confirmed cases now, much rarer than P. nolzi, but in Peninsular Malaysia, and also even very recently in West Kalimantan as well. Importantly, we have been thinking about the presence of other even rarer monkey species as well. There's some interesting studies from Malaysia and also in the recent paper from West Kalimantan where other species such as P. inui and P. courtneyi, P. simiovali may also be present, although some of the detection tools for these, given that they're often very, very low level parasitemia, still need some more definitive evidence. So thinking about the ecology of transmission and the impact on our understanding of zoonoses, particularly for monkey malaria. In response to a lot of human-related land use change and deforestation, we are seeing some of the mechanisms in terms of, for example, a related lack of lower biodiversity. So, for example, maybe the less presence of dead-end hosts with small mammals, and more encroachment on previously forested areas by humans, increasing the interaction between the monkeys and the mosquitoes. All of these features imply an increased risk for P. nolzi infections in humans, and really it highlights that our understanding of transmission needs to take into account not just human health, but also the environmental drivers and the monkey and wildlife factors as well. So this is sort of a classic and a fantastic example of a One Health understanding of disease transmission. This is a really nice schematic to highlight how some of the mathematical modelling shows that intact forest areas where there's high transmission in the monkey hosts, humans going in and out of those areas are at highest risk for spillover infections from the zoonotic parasites. To really highlight the degree of deforestation and land use change across Southeast Asia, particularly over the last 20 years, this shows that, in many of the areas where the monkeys and the mosquitoes have had to respond to this quite marked decrease in their intact habitats. This manifests, for example, in Malaysia, where it's been nicely shown that the incidence of P. nolzi infections in humans overlaps with those areas of historical forest loss. An even more detailed study by Kim Fornesi demonstrated how different environmental variables such as forest fragmentation indices and the proportion of pulpwood and oil palm impacted on the seroprevalence of P. nolzi in areas of Malaysia. Importantly, like a lot of these environmental variables are more predictive at different spatial scales. So in terms of the mosquito vectors, the Anopheles leucospirus group is the primary vector for P. nolzi. It's traditionally a forest and a forest fringe mosquito. It mainly feeds outdoors, but is a very opportunistic biter. So if it will feed upon monkeys, but if humans are around or other animals around as well, it will feed on them as well. in the past, they've had a preference for biting higher up in the canopy, but more recent studies have shown that they are biting at ground level as well. What about some of the other groups within the Anopheles leucosphaerus group? The Hackery and Rapara subgroups are known to almost exclusively still feed on monkeys, so these are thought to be unlikely vectors. And more recently, some of the other vectors actually outside the leucosphaerus group, which would have important implications for the distribution in the at-risk populations. So, for example, Anopheles sondacus has had P-Nolzi DNA confirmed from mosquitoes. However, sporozoites haven't been identified in these infections, and it's still the jury's still a little bit out about how whether they're an incriminated P-Nolzi vector. Importantly, when we look at this map, this really just highlights the paucity of information and studies out there, specifically looking, one, for the presence of the potential zoonotic malaria vectors, but also the lack of understanding about which species and where and whether they're infected and can transmit P-Nolzi or not. Again, just to highlight that a lot of the WHO indicators for how we evaluate the efficiency and the behaviour of potential zoonotic malaria vectors is really unknown for all the major mosquito species. Importantly, we know that in areas of Malaysia, the most common and most efficient P-Nolzi vector there, Anopheles balabacensis, has adapted to primarily bite outside and much earlier in the evening. For example, in response to insecticide bed net use, that's not really indoors biting at night time is not an issue anymore. So a really nice analysis where both human movement patterns overlaid with some of the mosquito catching data highlighted that even though abundance, for example, was higher in the forest, inside the forest, the highest risk of infection remained at the forest edge in some of those agricultural fringe areas. When thinking about the monkey hosts, there's been limited studies really looking at the adaptation from the monkeys in terms of their behaviour to deforestation. However, one GPS colouring study demonstrated that in response to deforestation, macaque troop home range size, movement speeds and habitat use really changed after, for example here, conversion to an oil palm plantation. However, sometime after that, the behaviour did sort of equilibrate and, new habitat was sought in response to that land use change. And in terms of human factors, a large case control study that we conducted in Malaysia, looking at individual risk factors associated with P-Nolzi infection, really demonstrated that male adult agricultural workers, working at the forest fringe and away from the house were the demographic group most at risk. There was still evidence of peridomestic transmission. However, we really highlighted that even other high-risk activities such as clearing vegetation in the last 4 weeks and previous palm oil plantation work were the highest risk factors. And I think that really gets me on to, the last couple of slides now. We're starting to conceptually think about how we understand and evaluate the potential risk of zoonotic malaria transmission, particularly in Indonesia. And the large ZooMel project, which we're doing with a number of key Indonesian collaborators who are leading the work, particular Inka Lubis at USU in North Sumatra and Rintis Novianti in North Kalimantan, where we're combining a lot of different approaches such as the human molecular and serological surveillance, really trying to understand and catch mosquitoes to understand the vector behaviour in certain habitats. We're also looking at macaque camera trapping and audio recording to really understand where macaques are occupying different ecotypes and at what times. And then some of the spatial analysis to really try and understand both from a quantitative and also from a social science perspective, what are the drivers in terms of human behaviour and land use, particularly for agricultural activities and forestry use in these areas. It's a One Health multidisciplinary project. The whole point is to have no sort of independent research silos and really co-design the study components. We, are very keen to include and engage really relevant stakeholders, particularly across, multi-sectoral government departments, in addition to some of the commercial large-scale plantations, as well as farming smallholders as well. We really want to show that there's mutual benefit both from a wildlife perspective, from an agricultural perspective, and a human health perspective. So thank you very much for listening, and I hope you enjoyed it. And again, thanks for the opportunity to talk today. Big thanks to many of our wonderful partners and friends who are working, that we're working with, particularly across Indonesia now. Thank you.
Thank you very much, Dr. Matthew Grigg, and it was a very nice talks, and I have highlighted several things here that Plasmodium knowledii can be cross multiple host families, including human, and it is already recognized as circulating in the many parts of Asia, including Indonesia. And the most important is that this P. knowledii is prone to be misdiagnosed because of the properties of this malaria, of this parasite. And One Health approach need to be applied to understand and the transmission and also the emergence of this disease, especially because of the changing in the human habit and also deforestation, and also the change of the vector and reservoir also has a characteristic. So we have already spent for 1 hour to listen to the talks from 3 speakers, and it is true that One Health is important in many areas, including in the tropical medicine. And there are challenges for not only to embrace stakeholders to understand, but also to, more important is to implement the One Health to this field. And thank you for very much for the speakers, Professor Wiku Adisasmito, Professor Roger Frutos, and Dr. Matthew Grigg. And also thank you very much for all the participants thank you for joining and also your attention. And please allow me to close the session and back to Dr. Eta as Master of Ceremony. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.